to those of you who are here in the room and to the people who are joining online, who that probably sounded really terrible for. Today, we're really excited to welcome Dr. Amanda DiNofia, who will discuss challenges arising for academic sponsors of early phase high-risk clinical trials in pediatrics. I'll introduce you in just a second, but we have a few announcements to start. So this is the first reps lecture of 2025. Starting from today, we are going to be holding reps on the second Tuesdays of each month. So make sure that we've changed that on your calendars. We're not on Wednesdays anymore. In February, we're going to be joined by Kate Spector-Baghdadi from Michigan, who's going to be talking about enabling genetic research with diverse ancestral populations. And you can see the full list for the spring semester here and on our website. We do have a very long list of prospective speakers, both internal to Penn and CHOP and external, but we always love to hear from you about people that you would like to see in this slot. So please share suggestions for people that you'd like to see here, especially people from diverse backgrounds and who would contribute diverse perspectives to issues relevant around research ethics and policy. We do have various online resources related to research ethics from our department. This is a series of short courses available for free continuing ed credit, some of which can be used to satisfy Penn and CHOP's human subjects training requirements. And then this is a new slide that our online education team has asked me to share with all of you, that there will be a behavioral economics workshop taking place from April 7th to May 2nd. You can snap a picture of that QR code, and the registration date is January 31st. And then just a quick note of thanks to all of our reps co-sponsors shown here, and thank you to Lisa Bailey for helping us get this together. So it is now my pleasure to introduce you. Dr. Amanda DiNofia is a pediatric oncologist in the sections of cellular therapy and transplant and hematologic malignancies at CHOP. She's also assistant professor of clinical pediatrics here at PSOM. Dr. DiNofia is responsible for clinical trial development and study startup and conduct for cellular therapies in pediatrics. She also serves as Associate Chief Clinical Research Officer for the Research Institute at CHOP, where she's built infrastructure that helps lead the efforts for CHOP and CHOP investigators to serve as sponsors for high-risk early phase clinical trials, which is what we're going to hear more about today. Just a word about questions and discussion. Dr. DiNofia has said we can interrupt her throughout, so please feel free to do that. Those of you online are welcome to post questions in the chat, or if you raise your hand, I can call on you that way. And are we recording this session?
Yes.
So this session will be recorded and that will get posted on our department's website. Okay, please take it away.
Thank you guys so much for having me today, especially to Holly and Steve and Lisa for having me. This is really exciting. We've been engaged in this work now for the past 5 years. And usually when you talk about kind of FDA regulations, people's eyes glaze over because if you really are having trouble falling asleep in the middle of the night, pick up a regulatory document and that will do the trick for you. But I'm going to try to make it really interesting today, kind of supplement with some clinical exciting data, but please interrupt. I'm Italian American. I like it when it's like saucy and we're kind of interacting back and forth. So please interrupt with questions as they come, and then all the feedback afterwards to make this less FDA reg eye glazy, I would be happy to hear. Okay, so what we're going to talk about today, first we'll start with just some rationale and mechanism of CAR T cell therapy. We are here at CHOP and Penn. This is like our birthright to know about CAR T cells. But just to make sure it's a level playing field, I'll give you some background about CAR T and its development and kind of the early data that we saw in pediatrics that got the world really excited. And then we'll talk about kind of what is the regulatory burden of opening up and holding the INDs for these really high risk clinical trials in pediatrics, knowing that there's a different level of rigor to test these therapies in kids. We'll talk about the different approaches that we have had to IND applications, depending on the trials that we have had open, and try to kind of look at it through that lens. And then just kind of lessons learned in the last 5 years, knowing that we're kind of at the early stages of developing this sponsor program at CHOP. And I can tell you kind of what we have learned along the way and some pearls, and I'm sure we will be adding to that as the years pass by. Okay. So this is, you can't like listen to a BALL talk on pediatrics and not see this graph. This is something that we're really proud of. So if you look at this survival curve, these are survival on the y-axis and then years since diagnosis on the x-axis. And then these different lines represent really kind of about 5 to 10 years of time. And you can see if you were diagnosed with BALL, which is the most common cancer in children, before really 1960, it was a death sentence. And you can see these kind of big incremental improvements. We were from multi-agent chemotherapy, the introduction of prophylaxing the cerebral spinal space, led to big improvements, and then really kind of multi-institutional trials to share data, because even though BALL is the most common cancer in kids, it's still a rare disease. Thankfully, doesn't look like that when you're on the third floor next door, but it is. There's about a little over 3,000 kids in the US will be diagnosed with BALL each year. But now, when we meet those families in the emergency room, we can say to them, for all comers, there's about 90% overall survival for those children newly diagnosed with BALL, which is an amazing victory when you look back to see earlier generations did not have that same success rate. But since 3,000 kids are diagnosed each year, knowing that 10% of them won't be survivors, who are those kids? And I will talk to you about them now. So they're typically two categories. One is patients who are refractory to our traditional chemotherapy. So we call that refractory disease. This is a survival curve of disease-free survival on the y-axis and years from diagnosis on the x-axis. And MRD is residual disease. So can you still measure leukemia? And responsiveness to chemotherapy is really the most important prognosticator in BALL. And so after 1 month of therapy, even though therapy is 2.5 years long, we meet them in the ER and we say, we're going to treat you for 2.5 years. That causes a lot of anxiety. But we say that after 1 month, we want you to be in remission. The rest of that time is really to maintain that remission. And we know that if you're not in remission after that first month of therapy, that the prognosis really becomes much worse. And this is a curve that shows the disease-free survival for those patients who do not achieve remission after the second month of therapy consolidation. And that's this red circle here. So you can see that the 5-year disease-free survival is less than 40% for patients who still have disease after the second month of chemotherapy. And so we need to do better for this population. The other big set of patients who don't do well with BALL is patients who have relapsed disease. So they achieve a remission initially, but then they have the disease come back. The curve on the left is for first relapse. We know there's different ways that we can prognosticate how patients will do after first relapse. And one is their time to relapse. So the earlier you relapse from your initial diagnosis, the worse you do. And you can see that patients who have an early relapse have a less than 30% overall survival. For patients who have multiply relapsed disease, this is the curve on the right. This shows that kind of 10-year survival is less than 10% in this patient's population with multiply second or third relapse of disease. So these are patients that have a really poor prognosis, and these are in that 10% of patients that we really need to improve outcomes. All of these curves that you've seen so far are before immunotherapy, the dawn of immunotherapy. So here we are with CAR T-cell therapy. So you probably have seen this picture or a picture like this in the past. It's hard to kind of escape even the lay literature without hearing about CAR T-cell therapy. So this is a schematic of what a CAR T-cell is. And so this is a CD19 CAR, which was the first successful CAR. And so the one that was often discussed. And this is the one that was developed here at CHOP and Penn. And so patients' own T cells, so they're T cells from the patient who is going to receive the therapy, are collected through an apheresis machine, and then they are manufactured in the lab to, let's see if I can, lovely, to express this CAR. And what a CAR is it has a monoclonal antibody fragment that is targeted towards an antigen or a protein on the cell of interest. And the CD19 is a really, kind of perfect antigen in which to target because it is only on B cells. It's not on other healthy, normal cells in the body. So you think about chemotherapy and how toxic it is for patients to receive. It's because chemotherapy just targets rapidly dividing cells. So patients get hair loss and nausea, vomiting, and other organ toxicity because you have lots of normal, healthy, rapidly dividing cells in your body. But CD19 is only on B cells. So it only takes out those cells of interest. And so when it meets its target, so patients get those T cells collected, they get manufactured in the lab, and then the patient comes back to clinic and they get these CAR T cells now infused. And I just infused cells at 11 a.m. So we do this a few times a week. And even though we've been doing this for many years, it's still exciting to think that you are infusing back a living therapy into a patient. So versus the chemotherapy that stays in your body for a few hours to days, depending on how it's metabolized, this CAR T cell is a living therapy that should provide ongoing surveillance from anything that with a CD19 antigen on it and destroy it, and then give itself a signal to multiply itself so that it is maintained continually in some patients for many years. So that's really exciting that CAR T cells have the ability to be really effective against BALL, but they also come with some toxicities. So it's important to kind of have that primer and know that. So the acute toxicities that we can see after CAR T cells are kind of in three major categories. One is cytokine release syndrome. So these are immune cells that you're infusing back into the patient. And so when they meet their target and destroy it and multiply, like I said, it was just very important for the CAR T cells to be effective. They have to go through that process. When they do that, they release a lot of cytokines into the bloodstream. And those cytokines are what cause a lot of the symptoms that we see after CAR. So when you have kind of flu or COVID or strep throat, the kind of the fever that you get, the kind of body aches and pains, that's actually not related to the virus that you contracted. It's related to your immune system's response to that virus. Well, it's similar in CAR T cells, that this manifestation of the CAR T cells working are these clinical symptoms. So CRS is kind of the blanket umbrella that explains, kind of describes most of the toxicities we see after CAR. The CAR T cell toxicities that are specific to the central nervous system is called immune effector cell associated neurotoxicity, which is a ridiculously overcomplicated name that we say, we call ICANS because it's much easier. And then if you thought that wasn't complicated enough, a third category is immune effector cell hemophagocytic lymphohistiocytosis syndrome, which is IEC-HS, which we'll talk about later in the talk. So these are kind of three major categories of toxicities that you can see. Typically, you're going to see them within the first month or maybe 6 to 8 weeks in some instances for ICHS after the CAR T cell infusion. So they can be quite serious toxicities, but they are often mostly self-limiting and they are acute versus this 2.5 years of therapy that we talked about, which was the standard of care for BALL. And then the two chronic symptoms or side effects that you can see after CAR T cells are hypogam. So your B cells are what make antibodies to help you fight infection. And I just told you that the CAR T cells will take all of your B cells away, leukemia B cells and normal healthy B cells. So we have to replace that in the immune system. And then a lot of patients will have low counts for some period, weeks to months after the CAR T cell infusion, which we can largely manage, but is worth noting as a chronic side effect. So just kind of quickly to get everybody on the same page about these toxicities that we can see. I already told you that the cytokine release syndrome is related to the T cells expanding to doing their job. So it is not a toxicity that typically we are upset about when a kid gets admitted for fever. We feel a little bit like masochists because we're like, okay, they're going to fever, but this is the CAR T cells working. So we will take it as long as they're falling on more of this kind of mild end of the spectrum. CRS is on a spectrum of severity and kind of more severe can be multi-system organ failure, but on the more milder end can look like the flu. It typically occurs within 2 weeks of CAR T cell infusion and can be a little bit later with other newer constructs. And is where you fall on that severity scale is entirely related to how much disease you have in your bone marrow at the time of infusion, which makes sense, right? I said it's related to T cells expanding. The more work the CAR T cells have to do, the more cytokines that are released, the more severe the clinical symptoms can be. The ICANS, or the CNS-specific toxicities you can see after CRS, also are on a spectrum. They can look anything like confusion. I had a patient who was studying for the GRE while she was getting her CAR T cells, which was pretty ambitious. And she said that she kept reading the same page over and over again, like she could not compute the information. So that's obviously on the more milder end, and somebody who was really astute, who was able to kind of name that really kind of mild symptom. But on the more severe end, we can see true encephalopathy. Patients lose their ability to speak or recognize their family members. And then kind of the most severe is seizure or spinal cord injury or cerebral edema. These are very feared toxicities. And we understand less about the mechanism of ICANS than we do with CRS. So there's lots of investigation to kind of do better here and predict which patients are going to fall onto what end of the spectrum here. And management is really steroids and supportive care. And then ICHS is a newer emerging toxicity, which we will talk about later, but hemophagocytic lymphohistiocytosis is a syndrome, or HLH is a syndrome that you can see in children or adults. That's a kind of a hyper-inflammatory syndrome. And we can see a particular signature of that HLH after CAR T cells. And it is independent of CRS. So patients typically recover from CRS and then a week or two later can manifest with new decline in their counts, irritation in their liver, and signs in their labs that they have kind of hyper inflammation happening. This is typically associated in particular with CART22 products. So we showed a schematic of a CART19, a CAR T cell that was targeting the 19 antigen on B cells. There's other antigens that can be targeted, and there's other diseases that can be approached in CAR T cell therapy, which we will talk about. But a CAR T cell that we've been developing at Chop and Penn for some time is one that targets the 22 antigen on B cells, and ICHS in particular is associated with that CAR. Okay, so this is probably some of you guys have seen these reports of this kind of early efficacy of CAR T cells. This was the first New England Journal brief report that came out. First author was Dave Porter, describing the early efficacy in 2 adult patients with CLL. The follow-up by Steve Grupp, describing the first pediatric patients treated with BALL and the first description of severe CRS and the management of that. The seminal paper to follow was the description of the first trial of CAR T cell therapy here at Chop and Penn that described 30 pediatric patients and a few adult patients. And this really kind of changed the game in BALL therapy and in CAR T cell development. There was other studies done at other institutes, both here at Penn in the lymphoma space, and then at other institutions describing other CAR T cell therapies that quickly followed. And then finally, not finally, there's still so much ongoing, but kind of the seminal paper that described the registration trial that went on to become support the FDA approval of first FDA approval of CAR T cells. So the first trial that we talked about that was published in that 2014 New England Journal of Medicine was called CHOP 959. And that was the first pediatric phase 1 trial of CAR T cell therapy that was really promising. And so Penn and Chop partnered with Novartis, which is a pharmaceutical company, to develop this product further and hopefully to FDA approval to allow better access for patients, and eventually led to this registration trial called Eliana. That, they had to demonstrate that, yes, you can treat patients with CAR T cell therapy when you have this state-of-the-art manufacturing facility on Penn's campus, but can you distribute this product that's manufactured at a few different manufacturing facilities around the world and ship those products and treat patients in different centers and still it be effective and still it be safe. And so that was what this registration trial was. It was at 25 sites in 11 countries, and Chop and Penn were really the clinical leads. And the outcomes were really incredible. So this was the first, on the left was the first curve that was published in that New England Journal paper in 2018 that showed that EFS at 6 months was greater than 75%, which, remember, this is in those relapsed refractory populations, which we saw the curves in the beginning of the talk that don't look nearly as promising as these do. This was a median follow-up of 13 months. And the follow-up study on the right here was a median follow-up of 30 months. To show that patients who responded 1 month after the CAR T cell infusion really largely had a durable response. and the median EFS among responders wasn't reached even at the 30+ month follow-up, which was really promising. So this went on to support the first FDA approval of CAR T-cell therapy. And that first FDA approval was in pediatric population, which is a real big victory for us pediatric providers because we almost never get a first approval in kids. And so this was a real source of pride. The approval was first in children and young adults for patients with second or greater relapse or relapse after bone marrow transplant, and that was in August 2017. Since that time, there have been 6 subsequent CAR T-cell approvals by the FDA. They are all using CD19 targets or BCMA, which is a multiple myeloma. They are all in adults. So we did well in the beginning with the pediatric approval first, but more work to be done to continue to allow better access for these CAR T-cell therapies for children across the country. We'll say at CHOP, 80% of the kids we take care of in the CAR T-cell space are not from Philadelphia. So patients have a huge burden to travel to access these therapies in early phase clinical trials. The patient that I infused at 11 o'clock, her dad had to get kind of a ad hoc job in New York to be somewhat close to them because they couldn't be here in Philadelphia for 5 weeks without income. So these things are real for patients. So this is what we work so hard to do. So there are continued challenges in BALL. So these are things that we should all feel proud about being here at Penn, that this is that Kymriah approval resulted from the work that was done here. But we can't sit on our laurels either, because there's still so much work to be done. You could see on those curves that even though they were much more promising than the first ones I showed you, there are still kids who relapse after CAR T-cell therapy. So and those are typically in the setting of the CAR T-cells don't last in the body long enough to provide that ongoing surveillance to prevent the leukemia from coming back. That's one instance. That's a CAR T-cell persistence problem. The other is that the leukemia in its infinite wisdom can get smart to the CAR T-cells and hide the antigen that's being targeted. So in particular, we call it a CD19 negative relapse that can happen after a CD19 CAR treatment, which is unfair, but well described. And so that is why there are CAR22 products, because patients who relapse with antigen negative disease need alternatives. There's also patients who, I told you this is the patient's own T cells that are being collected and manufactured into CAR. And some patients don't have healthy enough T cells to tolerate have enough T cells to be collected to manufacture a product, or healthy enough T cells after they've been battered with leukemia treatment for many years. And we're seeing that particularly in the solid tumor space too. And so there needs to be better work to get around that. There are investigational agents trying to look at donor CAR T cells, but there are challenges there. So there's also pediatric cancer outside of BALL. Clearly I'm biased because I'm a leukemia doctor, but there are other type of cancers in kids, and we have a lot of work to do better and give these kids CAR T-cell alternatives. So there's T-cell ALL and AML, acute myeloid leukemia, neuroblastoma, and CNS tumors in particular. CNS tumors are the second most common cancer in pediatrics. So there is a lot of interest to try to improve therapies in that space. And neuroblastoma, relapsed neuroblastoma is almost universally a death sentence. And so really trying to get better therapies for those patients. There's also pediatric disease outside of cancer that is very morbid, and in particular, lots of interest in autoimmune diseases, using CAR T-cells in that space, and really encouraging small adult data that has come out in the last year or two. So big push to kind of push that to the pediatric side. And then we talked about this amazing manufacturing facility here that we have at Penn, but that is expensive and hard to access for lots of patients. So is there ways to get CAR T-cells quicker and cheaper? So these are just this is a quick list. Somebody could give a talk just about challenges that lie ahead in CAR T-cells. So we're going to transition a little bit into kind of operationally how we make these CAR T-cell trials happen. And so we have kind of hopefully now a shared understanding of CAR T-cells, their promise, and what continues to need to be investigated. So historically, just for context, all of these trials, CHOP 959 was the one that I talked about, but many after that were trials that were supported by the Penn CCI. We are so grateful for that support that allowed all of these patients to be treated. These trials represent hundreds of patients who were able to access CAR T-cell therapy because of that support, which is incredible, and it allowed the development of many new CAR T-cell constructs. But about 5 years ago, there was a push within CHOP to see if, not to see, but to kind of develop the infrastructure so that we could support these trials administratively. So CHOP has had rich experience since 2012, when the first famous patient pediatric patient was treated with CAR T cells, rich experience serving as the study team. So investigators, principal investigators, and kind of study team activities. But it was Penn who held the INDs or were the sponsor of these historical trials. And there was this push to really develop that infrastructure here at CHOP. CHOP, in particular, the Division of Oncology, has served as sponsors for some of our bone marrow transplant clinical trials. But like Steve and I were talking about before this talk, those bone marrow transplant trials involve kind of manipulating the stem cell product that patients get as part of their bone marrow transplant. And the toxicities that we see in bone marrow transplant are almost always related to the therapy that they get to prepare their body for the transplant, and not that investigational product. So while that is a really important avenue to get patients the best and least toxic product, it isn't a high risk endeavor in the same way that CAR T-cell therapy is, which is why I wanted to show you all those toxicities to really prove that this can be really serious and toxic, albeit effective. And then CHOP as an institution has investigators that have served as both kind of sponsor investigator for certainly single patient INDs and smaller or lower risk clinical trials, but there had been no CHOP sponsor experience for really early phase, high risk clinical trials. And so that's what we were tasked with. But now to we have a level playing field from the clinical aspect, I just wanted to make sure we had that from the regulatory aspect as well. I'm kind of throwing around words like IND and investigators. And so just wanted to make sure that we all were thinking the same about these definitions. So a clinical investigation is an experiment in which a drug that's not approved or not approved for the proposed indication is administered or dispensed to subjects. And the patients who really, or the people who really lead those investigations, are the principal investigator and the sponsor. And a principal investigator is that person who supervises and often participates in the conduct of the clinical investigation. They are responsible to the patients. They protect the rights and the safety of the patients. And they report to their kind of big boss to make sure that they are doing their job is the Institutional Review Board, or the IRB. The sponsor is the big boss. They are the ones that take responsibility for and initiate the clinical investigation. So they're the ones that kind of empower the principal investigator with their role. They ensure that the investigation is being conducted according to the protocol through all different levels of oversight, which we'll talk about. And they report to the FDA. So that is their kind of person who's ensuring that the sponsor is conducting appropriate oversight. And so what is an IND? Well, an IND is an investigational new drug. This is not something I Googled when someone asked me to lead this program 5 years ago. Don't worry about that. I did not do that. I already knew that. But an IND, in case everybody doesn't know, is an investigational new drug. And an IND application is what is submitted to the FDA if a drug or a biological product, in this case, that was not previously authorized for marketing in the United States is intended to be used for the purposes of a clinical investigation. So that's a lot of words, but what that means is that you can't just decide to treat a patient with any construct and, make your own protocol and take care of patients, even in your best interest, even if you're, doing things kind of in a really kind of rigorous way. You can't do that without asking the FDA if that's allowed. There's strict regulations around that. And I think there's lots of kind of areas in the that people don't kind of understand that's really government regulated, that if you want to use a medication or a biologic, you need to ask the government if you can do that. And you need to provide really extensive justification to show why you are asking those questions and how you are going to oversee the investigation and keep the patient safe. So when you submit that IND application to the FDA, they have 30 days to review the application. And in those 30 days, they can come back and ask the sponsor team as many times as they please questions about this extensive application. But they have a 30-day timeline. And so what are the components of an IND application? Well, you have a cover letter to kind of plead your case to say why you think this is really important and interesting to study. The form 1571 is kind of where the sponsor signs the dotted line. And so that's kind of where they say, I'm going to be the sponsor and I'm taking responsibility. It has, of course, an introductory statement, the general investigational plan, which essentially I think of kind of like as the abstract of the IND, really kind of short form, summarizing all the data that the FDA will see. You can have an investigator's brochure that's depends on the application, and we'll talk about an IB later. The clinical protocol, that's really going to be the meat of the IND application to describe the protocol, what you're going to investigate. This is really going to dictate the investigation and how we'll go forward. The chemistry, manufacturing, or controls, or the CMC, which has really very detailed information about how the product will be made. In CAR T cells, that's manufacturing runs. You have to do some manufacturing, kind of practice manufacturing runs in your manufacturing facility to show that it's feasible to do and that you can make this CAR. For small molecules, that's, obviously a different, it's more kind of chemistry related. Then the pharmacology and toxicology is essentially all your preclinical studies. So did you study these CAR T cells in mice? Please describe all of those experiments in great detail. And then the previous human experience. So if this construct or a similar one has been used in humans before, the FDA wants extensive background data on that. So these IND applications can be greater than 1,000 pages in length. This takes months to develop and is a big read. So the FDA typically has teams that review different components of it. So when you're in that kind of question phase in that 30-day review period, you will get questions from the CMC team, questions from the Pharm Tax team, questions from the clinical team. Okay. So we were tasked with developing this infrastructure to serve as sponsor for these high-risk, early-phase CAR T-cell trials. We thought, since this was the first time we were doing this, we were going to work to transfer the protocol that had recently been opened under Penn IND and open it under TROP IND. That was the basket trial, which is a trial looking at CTL019 or a murine CART19 that is the same construct that went on to become Kymriah, the FDA-approved product. So we think of it as Kymriah is made at the pharmaceutical company, and this product is made at Penn's manufacturing facility. The way I describe it to patients is to say it's the same recipe, but made in different kitchens. So if I make an apple pie in my kitchen and Steve makes one in his kitchen, it might taste a little different even if we're following the same recipe, but it's generally kind of the same idea. So this was a construct we had lots of experience with, both on this particular construct and then when you think about it being so closely related to the commercial product. So lots of efficacy data, lots of safety data. So why are we studying it? Well, we're studying it in patients who don't meet the FDA indication. So you can only give CAR T cells to patients who have second or greater relapse or relapse after BMT because that was what the FDA approved it for. If a patient has a relapse in their CNS, which requires extensive craniospinal radiation, which is incredibly toxic to the developing brain, you can't just say, I want to give CAR T cells to this patient. The insurance companies, first of all, will say, no, this is quite expensive. But the FDA does not approve that. And so we are looking to study this construct in populations for which there is no FDA approval with a hopeful goal of one day maybe even expanding the indications from the FDA, but that is me being overly optimistic, but that's kind of the idea. So we were thinking this would be a good one to take ownership of first, since it's a bit lower risk as far as these risky things go. And it's already open under Penn IND. So Penn already did, sorry, I went the wrong way. This is fine. So Penn, we could cross-reference the IND that was already open at Penn. So Penn opened this and they made this 1,000-page document. And so there is something called cross-referencing, which allows you to reference someone else's IND that's already open. And what that does, it allows the sponsor for the existing IND to share this information with a partner, scientific partner, without having that scientific partner see all the information because it's proprietary, which is the way it goes. And that totally makes sense. And so the sponsor has to get, the existing sponsor, so the sponsor at Penn, a CHOP, would have to get a letter of authorization from the sponsor at Penn to say this was allowed. We can't just say, oh, I would like to cross-reference a trial open at the NIH. The FDA doesn't allow you to do that unless the NIH says, sure, Amanda and team, you can reference our IND. So that requires, you have to be scientific partners in order for that to happen. So Penn was super supportive of us developing this infrastructure and was super helpful in us doing that. And they, of course, were supported this granting of this LOA that allowed us to cross-reference this IND. So what that meant is that, So what that meant is that these kind of really onerous parts of the IND application, the CMC, the pharm tox, the previous human experience, we could just cross-reference Penn's IND. So what that said is that we submitted to the FDA these sections, because of course we had to write our own protocol and, summarize it and all of those things, but we didn't have to give them any manufacturing runs, any preclinical data. We didn't have to summarize the human data that Penn had already done. And we just said, please refer to this IND for those components. That's a cross-reference. And so we submitted the IND. This was our first IND submitted under CHOP in December 28th. 2020, and we got the study may proceed within 30 days. So that's the goal. They have 30 days to review it. They are motivated to allow the study to proceed in those 30 days because they don't want it to hang on, but neither do we. And so getting that study may proceed is a victory within the 30 days. So, and study may proceed is like a real buzzy FDA word and study team word. That's like, those are the golden tickets that then you have the IND. However, we had still lots of work to do. Because yes, you can write an IND and submit it to the FDA and survive the back and forth of their information requests, but then can you support the work of the sponsorship? And so I always say when we get the study may proceed, great, now we got to do the work. So we opened the IND, but we didn't transfer the protocol over to CHOP IND until we felt like we had the work done. And so what did that work entail? So this is all the things we had to build out at CHOP. We needed to build a regulatory team. So you need to know how to write an IND application. And then anytime you want to change anything in the investigation, you want to change the protocol, you want to make adjustments to the manufacturing, or there's a preclinical study that was done that kind of changes the information a little bit, anything that is done has to be formally amended to that original application. So you need expertise in people who know how to do that. Every year after an IND is submitted, that now becomes your IND anniversary, and you have to submit an annual report to describe the investigation ongoing. And so you need to hit that deadline, and you need to present that data in a really professional way in which the FDA wants to see it. So we needed expertise in knowing how to do that. Remember, I said there's all levels of oversight that the sponsor employs to make sure that the study team is conducting the study according to plan. One of those is the Data Safety Monitoring Board, which are independent. There are scientists or statisticians and investigators that are independent of this clinical investigation, but have expertise in the area that can provide independent review. And so that happens at different cadence for the study team and employed by the sponsor. So you have to be able to have expertise to impanel a DSMB, to write a charter, to task them with this responsibility. You need to facilitate relationship with vendors. So we have lots of vendors that we work with, in particular on the Penn side with the manufacturing facility and then the correlative science lab. And you need to develop an electronic data capture or an EDC. So everything in the protocol that you say that you are going to collect on the patient has to be systematically collected and reported in electronic data capture. And so you have to have the infrastructure and the expertise in which to build an EDC. And so I just highlighted here that this is the stuff that we had a lot of experience with, study team stuff. So developing a protocol, writing an ICF, applying through the IRB and maintaining IRB amendments. And these are really important things to know how to do and do well, but it was a whole other beast to take on the other responsibilities as well. Another layer of oversight is monitoring. So monitoring makes sure that the data that you're collecting and the EDC is accurate and according to plan per protocol, and that they make sure that all the investigators and the regulatory filings are appropriate. And you need to employ them and then write a data safety monitoring plan for these for this level of oversight. And then safety team. So we had a safety team, of course, as a study team. We had research nurses who were collecting adverse events and reporting them to the sponsor, but we needed to kind of up the ante, make sure that there was expertise in knowing how to report that and the timelines in which you need to report safety events to the FDA. And then some pharmacovigilance build out. So not only are we just regurgitating information to the sponsor, now that we are the sponsor, we have to look at the data systematically, make sure that we're not seeing patterns in data that should be worrisome. And then dose limiting toxicities, which need to be collected in a phase 1 trial, require a different level of scrutiny. And so that making sure that we had that kind of level of expertise. And then finally, lots of guideline development to spell out all this work that we were doing, because if you're ever going to be inspected by the FDA, they want to make sure that this is not just something on whim somebody says one day, that these are kind of systematic processes that are being conducted by the study and the sponsor team. So we had to kind of build a SOP and guideline library. Boring, but necessary. Okay, so the next steps after we build it all out, you can see that that took almost a year to do. And we were kind of doing that as we were applying to the, submitting the initial IND application as well. So it wasn't until September of 2021 where we transferred the protocol over. So I try to highlight when we did something for the first time. So this became the first CAR T cell trial now under CHOP IND in September 2021. So yay, we were very excited about that. We felt like we were ready to execute. But there's more work to be done, which I kept saying. So what's the next thing that we did? Well, we wanted to open a humanized CART19 trial for BALL, but manufactured on the Prodigy machine. So we had experience with the Prodigy machine, which is kind of an all in kind of one system way in which cells can be manipulated. We have experienced that on the transplant space. And there were other institutions across the country who had built CAR T cells within these Prodigy machines, but we had not done that on CHOP and Penn's campus. And so we wanted to build, instead of the murine CART19, which was the one that went on to become the FDA approved product, and that we were studying in the basket trial, we wanted to study a humanized CART19, but manufacture it in the Prodigy machine on CHOP's campus. And so the story, we always talk about like, what's the story about this kind of trial? The story for the FDA was that we have considerable safety and efficacy experience with a humanized CART19. If you remember back to that slide that I said, all the trials that kind of that Penn supported as sponsor on the CHOP side, we had a phase 1 and a phase 2. So we treated almost 200 patients with a humanized CART19, but manufactured at Penn's facility. So we were trying to make the case that we could do this, but kind of manufactured in a new way. But this was a new challenge because there was no cross-reference. There was no IND open at Penn making these CAR T cells in this way. And so we needed to author for the first time our own CMC section, our own pharm tox section, and populate the fairly extensive human experience. And just to kind of orient everyone, I've talked about this a few times, but the clinical cell and vaccine production facility at Penn, the CVPF, is kind of a super well-oiled machine. This is an outstanding CAR T cell manufacturing facility that has really been an example for programs across the country and world. And we're so grateful to have them as partners. But we were also looking to study, can we just do this in this little machine here, which had been done, like I said, at other institutions, and really make a case for that this technology could be used to make CAR T cells that were just as effective, that would reduce the manpower and allow potentially point of care CAR T cells. Now, we also are thinking about these bigger pictures, which I didn't put on the problems to solve going forward, but CAR T cells outside of the United States in countries that are not as developed as ours. Is there ways to bring CAR T cells therapies to those places? And the Prodigy machine is being used in country and not necessarily Prodigy, but systems like it in India and other countries to try to see if this can be expanded to other places. So we, the FDA bought our story and we got a study may proceed in July of 2022. Steve Grupp is the sponsor, Ali Lay, he's the PI of that trial. And this was the first all in-house CHOP CAR T cell study. So we were the study team, we were the sponsor, and we also had the manufacturing. And this was our first dose escalation. So this was our first foray into phase 1 with us being sponsors. So really kind of that level of safety data scrutiny with dose limiting toxicities. This was also our first and preferably our last placement on a temporary clinical hold. Unfortunately, we had a patient who had a grade 5 event, so it died during the investigation from high-grade ICANS, which was devastating for all involved. And also, we learned a lot from a regulatory and clinical trial space from that patient. The FDA very appropriately put us on hold to kind of investigate why that happened and to come up with plans to try to mitigate that risk going forward. And so this trial was on hold, which is the thing that the FDA does when there is a safety event on a trial, and did allow us to reopen.
Can I ask a question?
Oh, please.
Is it common to enroll people who have had those kinds of negative experiences previously, just to see like if this new antigen target works better? Just, yeah, how common is it that people who have had those severe toxicities then re-enroll in a trial for a different?
Yeah, it's a great question. So now that there's an FDA approved product, Kymriah, and patients can get that at their home institution, really most of our referral base is patients who failed Kymriah and are looking for an additional CAR T. And a humanized CAR T is, there's rationale that could maybe get around why a CAR failed in the past. So there is, more than half of the patients treated on this trial and others that I'll talk about are kind of retreatment. And so what that means is some of them have sustained toxicities from the prior CAR. What allowed this trial to get off hold was that we said we won't enroll patients who had high-grade ICANS toxicity on this trial going forward. But we know that these patients also will die of their disease. And so there's kind of that risk-benefit balance. You wouldn't push patients through undue risk. We wouldn't ever knowingly cause harm. So you wouldn't give someone a really toxic therapy just because they were going to die anyway. That's not a rationale that we use in pediatrics. I said that as an example of something we would never do. But that balance is tipped in a different way in these disease settings.
Sense.
Mm-hmm. So I think, what supported the trial coming off hold was this really kind of encouraging data that even though that there was that terrible toxicity, by and large in the study population, it was fairly safe. There were 2 high-grade ICANS. One was the patient that we talked about, another who had grade 3 ICANS, and 1 grade 3 CRS, really not other high-grade events, and that the relapse-free survival, so the outcomes for these patients are really promising. This is a median follow-up of 5 months, so early. This was presented by Dr. Leahy at ASH this past winter, or this past December, but really a very positive curve here. So we'll see how this plays out over time. The other thing was this manufacturing cost. So the marginal manufacturing cost per patient was less than $30,000, which is less than 10% of commercial product CAR T cells that are on the market. So another kind of compelling argument to continue this investigation and try to make a case for further growth in this space. So this is active and enrolling trial. Back to kind of the retreatment of patients who fail CAR T cell therapy. We talked about antigen negative relapse as being a risk with CAR-19 therapy. And so we wanted to study if you give 2 constructs at one time, does that decrease your risk of having antigen negative escape? This was a trial that was being done at Penn. So this could be a cross-referenced IND. There's previous human experience here in the adult setting. So this was kind of a more straightforward IND submission. And we got a study may proceed in December of 2022, and Regina Myers is the PI of this study. But always things to learn. So this was our first time administering 2 CAR constructs to a pediatric patient at CHOP, and this was a first in children study. And because there's lots to learn, this is where we learned this new emerging toxicity of IECHS. And we saw that half of patients experienced IECHS and a good percentage experienced really high-grade IECHS. This gave us big time pause on this trial, pause as an internal pause, FDA pause. That's also a FDA buzzword. We were not put on hold for this trial, but we kind of stopped ourselves and really looked back at the data. What is really encouraging is this is a highly refractory population. Most of these patients have had a history of bone marrow transplant, has had a history of 1 or more prior CAR T cell therapies, and so heavily pretreated population with early response being exceptional. And so we kind of did an internal pause and thought, how can we go forward? Because this feels really worth studying, but do it in a safe way. And what we have done is what we found that their CAR-22 proliferation of the CAR T cells was really robust and was likely driving this IECHS. And so we dose reduced 22. We restricted enrollment to patients who had a history of CAR T cell therapy. So patients who had alternative therapies, including alternative CAR T cell therapies, were limiting, really trying to get the kind of highest risk patients. And then we were treating 6 patients. We're treating our 5th now, and then kind of reevaluating the data and presenting that to the FDA and the IRB before proceeding. So we're learning.
Amanda, we have about 10 minutes.
Okay. So next was developing this CAR T cell in the neuroblast in the solid tumor space. And so I talked about neuroblastoma being a really morbid disease. So this was our first CAR T cell product at CHOP in the solid tumor space. And this is our first in human study. So there's no existing data in this space. We cross-filed the drug master file at Penn, which is an FDA submission that provides confidential information about facilities. Since these CAR T cells were going to be made at the CVPF at Penn, but had never been made before, we can at least cross-file the kind of background information that Penn submits to the FDA about their manufacturing facility, but we had to do our own CMC and Pharm Tox section. The drug master file supports an IND application, is not an independent section of the IND. And so talked about that this was kind of brand new authorship. Since we knew that this was first in human, which was a new foray for us, we requested a Type B meeting with the FDA to make sure that we had all the information that the FDA would be expecting. And so what is a Type B meeting? That's a pre-IND formal meeting that you have with the FDA to get feedback on your IND application. So you give them kind of a Cliff Notes version of the IND application and you ask them for feedback. Like, would this support an IND application? Is there enough manufacturing runs? Is there enough preclinical data so that you set yourself up for success? We got a study may proceed from the FDA in April 2023. This is active and enrolling, and this was our first in human CAR T cell study under CHOP IND. And then hot off the press, which this was, I kind of had the slides final, and then we got this information, so I added to it because it's exciting. We have been developing using the murine CART19. Again, I keep saying this over and over again, but that's the one that went on to become the FDA-approved product that we are studying on the basket trial for new indications. So lots of safety and efficacy data in B-ALL. We have been developing using that construct in lupus in pediatric patients. And it's a similar IND story that we told to the FDA for the Prodigy trial, saying this is a very similar construct of which we have lots of information, but just being manufactured on the Prodigy machine in a slightly different way. And so it did require CMC because we are manufacturing at CHOP, but very little Pharm Tox because it feels silly to study this in mice when we have all this human experience. So the human experience is essentially all the stuff we've had at CHOP with this exact construct, and then essentially can extrapolate to the commercial product since they're so similar. And then this became our first CAR T cell product at CHOP for a non-malignant indication. And on Friday, we got our study may proceed. And Caitlin Elgarden will be the PI for that trial. So look forward to seeing us executing this in the non-malignant space. So just the last minute or so, I just wanted to give some kind of lessons learned over this experience. So we now hold 5 INDs for pediatric phase 1 and 2 CAR T cell studies in pediatrics, and there's 5 on deck. And so what have we learned? One thing is that there seems to be a difference in the FDA expectations from an academic versus an industry clinical trial. So all the FDA guidance, which are the things I told you to Google in the middle of the night when you can't fall asleep, or the FDA guidance documents, most of all of them have in their title industry. So like industry guidance for X, industry guidance for Y. They don't speak to the expectations for an academic IND. But in our experience, this offers some flexibility for academic submissions. So in particular, for investigator brochures and Part 11 compliance, which are components of clinical investigation and IND applications certainly for industry trials, they have not mandated that we provide those as part of our applications. And Part 11 compliance is kind of a rigor for clinical data. An investigator brochure is a compilation of preclinical and clinical data that you continually update about a construct. Both of those things aren't necessary if you are hoping to engage the FDA about a new drug application. So hoping to develop something into an FDA-approved product. But really the expense for a lot of these things, in particular Part 11 compliance and the data that is the rigor of the data that is necessary to support an NDA, is more than an academic center alone could ever support. And so you really do kind of need an industry sponsor if you're moving towards an NDA. And so our thought always is that we are developing these early phase trials to hopefully develop really encouraging preclinical data that if it's promising enough to develop into an NDA, we would have to engage an industry partner and have a different approach for those subsequent clinical trials. The other is the first in child and first in human. So there's really kind of written CFRs describing the kind of rigor in which is necessary to support a pediatric investigation. And so we know that we have to be, the kind of data that the FDA wants and the strictness around inclusion criteria is much more kind of stringent in this when we're making a case for a first in human study in pediatrics. And so we often are asked to treat older patients first, even if they're older pediatric patients. So we're actually doing that on the lupus trial where we have to treat kind of older adolescents before we're able to go down to age 12. That's just one example. The other is the malignant versus non-malignant. So the risk tolerance is lower in non-malignant populations. So we talked about the patient who had the grade 5 event on the Prodigy trial and how these patients are going to die of their disease. That's not the same in lupus, although that can be a terribly morbid disease. We really had to make a different approach to our clinical protocol and our IND application, talking really much more extensively about the chemotherapy exposure that prepares the patients for the CAR T cells because this population may not otherwise receive chemotherapy, and really make the inclusion and exclusion criteria much more narrow to allow to be risk tolerant enough for the FDA. And then just a few kind of minor things is that we found that really developing user-friendly EDC that creates reports that make DSMB reporting and annual reporting much more streamlined is absolutely essential. We've gone through a few in the last few years. We also had to create a biorepository for sample storage because that became really expensive to outsource with all of this kind of clinical trial activity. And then finally, that having an excellent team that we have built over the last 5 years makes all the difference. So we have just a great group of people who support this work. The leaders behind this work have really incredibly been Steve Grupp and Richard Applins, both in the CRSO and the CTTS section, who have supported this build out. And Melissa Varghese, who's our regulatory specialist, who is just an amazing wealth of information and it really is largely responsible for this great success. And then, our credible partnership with the Penn team, both at the CVPF and TCSL, which is the correlative lab, and the CCI with their kind of mentorship as we develop this program have been absolutely essential to the success. So that's what I wanted to talk about today. Certainly open to any questions in the last few minutes.
So I know you were tasked with building out this IND clinical capacity at CHOP. Could you just give me a sort of high level what is the perceived institutional advantage of, okay, you've got bits of it, but perceived institutional advantage to CHOP of doing these rather than through a collaboration with Penn?
Yeah, so financial. Yeah, very expensive to outsource this support. And can people hear at home the questions? Okay. Yeah, so financial, definitely.
Oh, no, they said they couldn't.
Oh, they couldn't hear. So the question was, what is the CHOP incentive to develop the infrastructure at CHOP rather than, do what was working really well, which was having Penn support? And yeah, I said primarily financial incentive because it was a lot of money to spend to get that work supported, which was done so excellently, but just, feeling like we had the expertise to do that here. We just had to kind of get the push to do it. And then the other is to innovate. We really feel like that's a big part of CHOP's mission, as you all know, CHOP and Penn's mission, and to just kind of demonstrate that we could do that too, of course, is always part of it.
MMR online says there's a potential IP incentive.
Sure. Yeah, those are all kind of built into certainly clinical trial agreements for sure. Mm-hmm.
A follow-up question to that. Do you have any sense, so yes, it was expensive to be sort of subcontracting to Penn to do it, but
Mhm.
CHOP now has to like create all of the teams.
Yeah.
These are really expensive, like operations that
Sure.
you described. It's enormous.
Mhm.
Do you have any sense of like where those funds are coming from and
Mhm.
like how CHOP is supporting? the ability
Yeah,
to do
that's a
this?
good question. It is a mix. The hospital itself supports a lot of this work. They see the value in the CAR T cell innovation and the service line. So it's financially beneficial to the hospital when patients come from outside the catchment, and 80% of the kids we take care of are outside the catchment. So it's both financially and mission driven that for CHOP. And so they feed a lot of money into the program. In particular, the manufacturing costs are largely supported by the hospital, which we're so grateful for. It really allows this work to move forward because the manufacturing costs are really the bulk of the costs. And they also support some of the infrastructure within CTTS and the CRSO, just FTE wise. And then grant funding, research funds. So it's a mix. But without CHOP's institutional support, we would not be able to support all these trials for sure. And these are high enrolling trials too. Over 50%, over 50 patients are on the accrual for most of these trials that I presented, which for phase 1, 2 and pediatrics are large, but we're trying to innovate and learn and also give access. So it's a combination of those things.
We're out of time. Thanks very much.
All right, thank you guys.